Hi everyone, my name is Allison Mayle. Welcome back to DNALC Live. So today on DNALC Live, I'm excited to talk to you about forensic DNA analysis. The technique I'm going to talk about is not actually used very much in forensics anymore, but it's a way to introduce some important molecular biology, molecular genetics concepts and techniques, and it's kind of fun to still do them in the context of forensic science. So I'm going to start by introducing you to restriction enzymes and gel electrophoresis, and then we will use those tools to try to solve a made-up crime. But quickly, I just want to introduce you to the DNA Learning Center and DNALC Live in case you're not familiar with us or in case this is your first time tuning into a DNALC Live event. So the DNA Learning Center is part of Cold Spring Harbor Laboratory located in Cold Spring Harbor, New York. We also have sites in New York City and in Sleepy Hollow, New York. And normally we have students come to us on field trips and for summer camps, but right now, while nobody can come on field trips, we're presenting some of our lab materials online on DNALC Live. And you see at the top of the screen, I have our social media handles, so I encourage you to follow us there. Subscribe to our YouTube channel if you're not already so that you don't miss any DNALC Live events you might be interested in. All right, so I'm going to get started with our presentation. So the first thing I like to talk about when I do this lab in the class is what are some types of forensic evidence? And I'm going to list some here. You may be able to come up with some different ones on your own. You can think about them if you're watching live and you want to include them in the chat, feel free. But I'm just going to list out some of the most common answers I get in my class. So some types of forensic evidence that might be recovered at a crime scene are fingerprints. That's usually one of the most common answers I get. Blood, perhaps from a victim or perhaps during a fight. Fibers or things left from bits of clothing if someone got their clothes snagged on something or something like that. Footprints or shoe prints, so similar to fingerprints but left by somebody's foot or somebody's shoes. Bits of hair may be left at a crime scene. Other body fluids other than blood may be left at a crime scene. Weapons might be left at the crime scene, sometimes not, but maybe. And there may be skin cells, cells from somebody's hand and fingers if they weren't wearing a glove and they were touching a weapon or something else at the crime scene. So usually at this point, I also would ask, fingerprints is often the first thing people come up with. So just why do you think fingerprints are good evidence for an investigation into a crime? And usually I hear back, well, they're unique to us, right? So everybody has a different fingerprint. So we can identify whoever left that fingerprint behind. Now it's important to remember when we're thinking about forensic evidence that just because someone left a fingerprint or any of these other types of evidence in the list doesn't mean that they committed a crime. It just means that they were there. And that would be one piece of evidence that investigators would use in order to help figure out what happened. All right, so fingerprints are good evidence because they're unique to each person. Sometimes they might be difficult to recover from a crime scene because perhaps someone was wearing gloves, or also if as they were touching something, they moved their hand, the fingerprint might be smudged. It might be hard to actually get a nice clear fingerprint. So let's talk about some of these other types of evidence that I've highlighted here in red. Blood, sometimes hair, depending on how the hair was left at the scene. Sometimes other body fluids and skin cells. What do these things all have in common? If you think about it, these things all have in common that they might be a source of DNA. Now, like I said, for hair, it depends on how the hair was left at the scene of the crime. It has to have a little bit of that skin tag left on it to really recover usable DNA, but it might. And so if we were able to get some of these things, we might be able to identify DNA that was left by someone who was at the crime scene. And we're going to talk about DNA a lot more in just a minute. But think about why might we want to look at DNA from a crime scene? So where is DNA stored, first of all? If you think about the things that were on that previous list, they all have something in common, right? They all came from somebody's body. And DNA is stored in our cells. Most of our DNA is stored inside the nucleus of our cells. There's some DNA also in our mitochondria, But most of the DNA we're going to be thinking about today or looking at in a crime scene investigation comes from the nucleus. So almost all the cells in your body, with the exception of your red blood cells, have DNA in them. Okay, so we have DNA in our cells. What is that DNA for? Why do we have DNA? Usually students can tell me you get your DNA from your parents, but why do we have it? So DNA is instructions for making us who we are. It's instructions for giving us our traits, so what we look like, what kind of features we have, and it's also instructions for performing functions, things that our cells need to do for us to be alive and for us to do anything we want to do. So things like carrying oxygen through our blood, that's encoded in our DNA. Our DNA makes instructions in order to actually perform that function. All right, so DNA is in our cells and we need it because it's important for making us who we are. How much DNA do you think all humans have in common? If you download the worksheet on our DNALC live page, you'll see that this is one of the questions that you could think about before we proceed with the lab. And if you watched at least one of our other videos, you might have seen this answer actually written on the wall in our learning center. I'm obviously not in one of our learning centers today. I'm in my living room. But how much DNA do you think, if we looked at my DNA and your DNA, anyone who's watching this video, how much of it do you think looks the same? Well, in fact, 99.9% of our DNA is the same for every human. And this is kind of amazing, right? 99.9, that's a lot of DNA. That's exactly the same for all of us. But if you think about it, it makes a lot of sense because all of us as humans have a lot of shared traits and a lot of shared features, right? We all have eyes and a heart and lungs and a liver and kidneys, and all of our internal organs are for the most part the same. We all have skin. It might be a little bit different color, but we still have skin that's performing the same function of protecting us from the outside world. And so 99.9% of our features and stuff that are coded by our DNA comes from the same DNA. And then those little variations make us different, make us unique. So in forensic science, we're looking at that 0.1%, that little bit that's different. And what's really great about that little bit that's different is it's different for every single person, except for identical twins. If you're not an identical twin, your DNA in that 0.1% is different from everyone else on Earth. So we're more alike than we are different, but we can use those differences to identify whose DNA was left at a crime scene. All right, so let's talk a little bit more about DNA so that we're all on the same page as we move forward to talk about our lab today. So this little illustration here of DNA, you can see in the green, something labeled adenine, in the sort of light purplish pink color, something labeled thymine, in the darker purpley blue, something labeled guanine, and then in the kind of reddish color, something labeled cytosine. Now these are often abbreviated just with their first letters, A, T, G, and C. And together those are called bases. So those are actually making up the instructions to do all those things I was talking about before, to make you who you are. So if you think of them just as their letters, A, T, G, and C, you can kind of think about them like words. All right, so if you had a series of letters in one order, it might mean one thing. If you change the order of the letters, it might mean something else. Same thing with our DNA. If you change the order of the bases, they will give you a different trait or change the way that trait looks. All right, and then I just want to talk a little bit more about the outside of the DNA in this picture. So in this picture, it's labeled a phosphate deoxyribose backbone. Okay, that's kind of a lot of big words. So phosphate are these little groups, phosphate groups that have a little yellow highlight in the middle of them. They're part of the backbone of DNA, so the part that holds it all together so that your instructions aren't falling apart. It would be very hard for our cells to follow instructions if they were just falling apart. And then they're attached to deoxyribose. That's a big word. Now, if you think about that word, though, it ends in O-S-E. If you think about other words you can think of that also end in OSE, like glucose or fructose, you might realize that this is a sugar, or lactose if you watched our lactase lab on DNALC Live. So there's actually sugar in your DNA, a sugar molecule called deoxyribose. So what we can do is we can use a tool that I'm going to talk about called a restriction enzyme. And it looks for a particular series of letters, and then it breaks this backbone apart, which is pretty cool. Normally, we don't want that to happen inside of our cells because, like I said, it would be very hard for our cells to follow these instructions if the instructions were falling apart. But in this case, we want to use this as a tool to look at something in the lab. Okay, so as I sort of just mentioned, restriction enzymes, they're a type of protein, they're an enzyme, and they cut DNA up at specific sequences. So I have two examples here on the slide. One is called EcoR1 and one is called HindD3. Now, you might have learned in one of our other videos or in your biology classes or one of your classes at school that enzymes usually end in ASE or ACE. These enzymes are named a little bit differently, so don't be confused by that. It's just a slight difference for these particular class of enzymes, that they have a different name that's based on where they were discovered. So these come from bacteria. We're not really going to talk about that today, but that's where their names come from. What I do want you to notice today is if you look at where EcoR1 cuts, you see that the letters say GAATTC across the top. And then if you look at the bottom and you read backwards, so from right to left, you'll see that the letters again say GAATTC. And this is a pretty cool feature of many restriction enzymes, that they cut in these sequences that are the same forward and backwards. So they're called palindromes or palindromic sequences, just like a word in English or other languages that's spelled the same forward and backwards, like race car is a palindrome. These sequences are often palindromes. And so EcoR1 would cut up the DNA in between the G and the A on the top strand and the G and the A on the bottom strand. So it cuts both strands of our double helix. That's the name of the shape of DNA where we have those two strands of DNA twisted together. And it would separate them and actually cut the DNA into pieces. But it only cuts if it sees GAATTC. If it sees any other series of letters, it just leaves them alone. Okay, and then the same thing for HindD3, just to give you two examples so you can see that they're unique. HindD3 only is going to cut if it sees AAGCTT. All right, it's not going to cut if it sees any other series of letters. Okay, so what does this have to do with forensics? Well, since we know that these enzymes only cut in particular sequences, we can use them to determine if that particular sequence was present. So let me show you an example here. If you look at these series of letters, these bases, from two different individuals. At the top, we have person number 1, and beneath them, we have person number 2. And in both of them, I've listed out 37 bases in a row. And if you look, you can see that they're mostly the same. Okay, so just like we have most of our DNA sequence the same, this is shown here in this picture. It's not quite 99.9% the same. We're looking at one of those regions where there's some differences. But you can see that these two people have mostly the same DNA. All right, so let's imagine though that we cut this DNA up with EcoR1. So I wrote there at the top, you can see EcoR1 enzyme cuts GAATTC, cuts between the G and the A. Now, just for the sake of simplicity, I only listed one strand of DNA, but remember DNA is double-stranded, so there would be another string of letters right underneath each of these letters that pair up. A always pairs with T and C always pairs with G, but let's just look at them as a single strand right now, just so we can kind of think about what would happen if we cut both of these people's DNA with EcoR1 that cuts GAATTC. You might have noticed already, if you've been looking at this for a few seconds, that in person number 1, just a few base pairs in, we have ATTC and then we have GAATTC. So that's going to get cut by our enzyme. But we don't have that sequence in person number 2. After our ATTC, we have GGATTC. So that one's not going to be cut by EcoR1 just because of that one little change from A to G. So I have this illustrated on the next slide. Just put little blue lines anywhere where this DNA would have been cut by EcoR1. Okay, so make sure I didn't make any mistakes. Take a look, see that I cut only where it says GAATTC and not any of those other sort of imposter sequences that look similar. All right, so what does this do for us? The DNA is going to be cut up differently, And then we can actually look at those pieces of DNA. We call them fragments. So we can look at the fragments of DNA. And you can see that in both of these cases, you'll end up with 3 fragments of DNA, right? Imagine if you took a ribbon and cut it twice, just like we've cut our DNA here twice, you'd end up with 3 little pieces of ribbon. But look at those pieces. On the far right side, the pieces look like they would be exactly the same, right? They would be the same size. But then in person number 1, we have this really big piece in the middle, and then another small piece on the left. Whereas in person number 2, we have that small piece on the right, but then we have 2 kind of medium-sized pieces to the left of that. Right, so we can actually see how big these pieces of DNA are. We're going to talk about that in just a minute. And hopefully that can help you see how we would be able to tell the difference between these 2 people without even knowing what the letters are. So here I've shown you the letters, but even if you didn't know what they were, you would be able to look at those pieces and see that they were cut up into different sizes. All right, so let me give you one more illustration of this that I hope will make it clear. So this time I've illustrated the restriction enzyme sites with these little scissor diagrams. So at the top, we have a piece of DNA evidence. So this is where our forensics comes in. Imagine that we took some blood or something from a crime scene. And we took the DNA out of that blood and we cut it up with EcoR1 as an example. We see where the EcoR1 cut is in those 3 different spots where you see scissors. Now, when we do a crime scene investigation, which we don't do at the DNA Learning Center in real life, but just we as a general statement, we always want to compare to the victim. If there was a victim of the crime, we want to make sure that we're not looking at their DNA. It might be helpful for us to know which DNA was theirs at the crime scene, but we also want to make sure that any piece of evidence that came from the victim, we don't try to match it to a suspect, because if it came from the victim, it didn't come from one of the suspects. All right, so in this case, you can see there is one cut site that matches up there on the left, but then the other cut sites are different. So this victim's DNA doesn't have GAATTC in the same spots as our DNA evidence. So did our evidence come from the victim? No, in this case, it did not. So now we could compare it to some of our suspects. All right, so if we look here at suspect 1 and suspect 2, which one do you think matches our DNA evidence based on this picture? Okay, so suspect 1 looks totally different, right? It didn't get cut the same number of times and the cuts are in totally different spots. But how about suspect 2? Suspect 2 matches the DNA evidence exactly. So we would see that it has the same number of fragments and the fragments would be the same size. So this suggests that this particular piece of evidence came from suspect 2. Now remember, just because a piece of evidence came from a particular suspect doesn't mean that person committed the crime, but this would be one piece of information that investigators would use to try and solve the crime, to try to figure out what happened. So we would say this piece of evidence came from suspect 2. All right, but how do we actually see these little bits of DNA? So I've already cut up DNA with a restriction enzyme for you. And we need a way to see it though, because if you just look at a tube of DNA, I didn't bring a tube over here, but if you just looked at a tube of DNA, it's gonna look exactly the same before and after we do the restriction digestion. That's what it's called when we cut up the DNA. So we need to do a special technique that allows us to see what size DNA we have. And that technique is called agarose gel electrophoresis. All right, so let's break this down by word. So agarose ends in O-S-E. We talked about this a couple of minutes ago. Do you remember what kind of molecules usually have names that end in O-S-E? Right, these are sugars. So this is actually a sugar that comes from a seaweed called agar or agar-agar. And we use that in this technique. And we use that sugar to make a gel. And you can think of this gel as being kind of like Jell-O. I'm going to show you an example gel in just a minute. So it's not like hair gel. It's not super goopy. It's a little bit more solid than that. So we make a Jell-O-like gel out of our agarose sugar and some other chemical buffers just to make sure everything stays exactly how we want it. We're going to use this gel for a technique called electrophoresis. Okay, so the first part of that word, electro, you probably think sounds like electricity. And that's exactly right. So this just means we're going to apply an electric field. All right, so we're going to use electricity. Now the part that says phoresis, that one's a little bit trickier. It's not as common in English, but what it means when you have the word the ending phoresis at the end of a word is to migrate or to carry. So we're going to use electricity to migrate something through our jello-like gel that's made out of seaweed sugar. What do you think we're going to migrate through this gel? Well, the thing we've been talking about, our DNA. So we're going to migrate our DNA fragments through the gel in gel electrophoresis. Okay. So before I talk about loading, I just want to show you what the gel looks like. So I'm going to stop sharing my screen. Let's see. And what I'm going to show you here is an example gel that I made. Let me take it off of the tray. So this is what it looks like. It's just kind of like Jell-O, kind of like a Jell-O jiggler, actually, if you've ever had those before. And what you can hopefully see is that there are little wells, little pockets here in the top. All right, so we made those because when we put our gel together, we use this thing called a comb because it looks like a comb that you would use for your hair. It has these little teeth. And as the gel solidified, so it's just like making Jell-O, I heated up the agarose and poured it into this little mold. It had something on the end before to stop the gel from running out of the ends. And I put this little comb on it so that anywhere where there's a tooth in the comb, the gel would leave a little indentation, a little pocket. All right, and those little pockets are where we're going to put our gel, our DNA, excuse me. They're in the gel. We're going to put our DNA there. So I'm going to start sharing my screen again. Whoops. Maybe. Here we go. Share screen. Okay. So I'm going to share my screen again. And if you couldn't see the comb before, I might have held it up too high. Here, I'll hold it up again so you can see me in the little top corner. That's what the comb looks like. Okay. So now the comb left these little pockets, and they're drawn here as little blue rectangles. And usually when I do this in a classroom, I have the students have some practice lanes on the outside. And the picture of the gel I'm going to show you in a few minutes is actually from a class that I taught recently. So you might see some lanes where there's a little bit of dye, but no DNA. That's because that's where they were practicing. And then in the middle of the gel, they have their different evidence, which is listed here as E. And I'm going to show you the crime scene in just a minute so you know where those evidence pieces came from. And then again, we compare it to the victim so we can rule out their DNA. And we compare to two suspects in this particular case, S1 and S2. So their DNA samples are going to be in the middle. So I had cut up the DNA with the same restriction enzyme for all of the samples. And then I also put in the DNA some dye, a special dye called cyber green that allows the DNA to glow when it's exposed to a blue LED light or a UV light. Otherwise, we still wouldn't be able to see the DNA. I'm going to talk about how we separate the fragments in just a minute. But what I want you to notice right now as well, in the bottom of the gel picture, there's a little triangle. That's representing a pipette tip going into the well and loading the DNA into that spot. So I'm going to show you how we use a pipette in case you've never used one before and you'd like to have some idea of how it works. And then I'll show you a video of loading a gel, and then we'll talk about our crime scene. So I'm going to stop sharing one more time. Oops. And I am going to talk, before I talk about the pipette, I want to talk about something like this. Let me just do this. All right, so even if you've never used a micropipette, which looks like this, you may have used something like this before. A little dropper pipette, sometimes called a transfer pipette, or some sort of eyedropper. So a lot of times I start my class by asking the students, if I wanted to use this dropper to transfer some liquid, imagine I had some liquid in this beaker, right now it's just air, but, and I wanted to transfer some liquid from this beaker onto the floor. I don't know why, but just for fun. What would I do first? Because this works exactly the same way as the micropipette, but the micropipette is more precise. So what I would do first, right, is squeeze the bulb, and that lets the air out of the pipette. And then I would put the end of the pipette into the liquid and release the bulb. And that lets the liquid replace the air that I've squeezed out. Right? And then I wouldn't squeeze it yet, I would just remove it from the beaker. And if I wanted to dump it here on my floor, I would squeeze the bulb again. All right, so that seems a little bit silly, but like I said, that's exactly how the micropipette works. And so I think it can be helpful to think about something you're already familiar with when you're using a new tool. All right, so when we use our pipettes, we also don't want to mix up our DNA samples. If I put the end of this pipette here into the DNA sample from the evidence, and then I put it into the DNA sample from one of our suspects, what would happen? They would get mixed up, right? They would get contaminated. We don't want that to happen. So we use these things called pipette tips. I'm gonna hold it up to the screen here so you can see there's little clearish white things inside of here. These are called pipette tips. This is just like if you go to the doctor. I'm just gonna put a pipette tip here on the end of my pipette. If you go to the doctor and they look in your ear and they put a little plastic coating, right? A little plastic cap before they look in your ear, and then they change it before the next patient. That's what this little pipette tip is for our pipette. And so I'll just show you here with some practice loading dye. So this is just a purpley colored dye. So what I would do, just like with the other pipette, instead of squeezing the bulb, I'm gonna press down on the plunger. So I'm gonna press down. And when you press down, you'll see there's one stop here, and then you can press down harder, it goes all the way down. That's called the second stop. So I'm gonna press down just to the first stop. That's gonna let out a precise amount of air, a precise volume of air. Now the reason we wanna do this is when we're comparing our DNA samples, we wanna make sure that we're comparing the same amount so that we know that no differences are just because we've put a different amount of DNA into the gel. And also when we're setting up our restriction digestion, we wanna put the same volume of DNA in, we wanna put the same volume of restriction enzyme in. We wanna have everything exactly the same, except for who the DNA came from. Okay, so I'm gonna press down to the first stop. Then I'm gonna put my pipette tip into the liquid, and I'm going to release my thumb upwards, just like releasing the bulb. And then what you can see is I have this little bit of purple liquid in the end of my pipette tip. All right, and then I'm gonna put it back into the same tube, but you would put this into your gel or into whatever tube you were transferring into. And you press all the way down to the second step to get your DNA out of the pipette tip. And then you would throw that old pipette tip away and get a new pipette tip for every single sample that you were running on your gel or that you were setting up to do an analysis. Okay, so what you would do instead of just putting the liquid back into the tube like I did right now is you would have your gel set up in this little chamber, which I'm going to show you here. We have lots of different kinds of these at the Learning Center. This is the one I brought home with me. So you can see now the gel is just sitting inside this little plastic chamber. And there are some electrodes on the end. So that's how we're going to use our electric current later. So you can imagine, and I'm going to show you a video of this because it's hard for me to be able to show you very clearly on the video, that I would have pipetted the sample right into those little wells inside of this gel box. And then we also have to put some liquid over the gel so that the electric current will actually flow through the gel. And again, I'm going to talk in just 1 second about how the electricity affects the DNA. But I'm just going to go back to sharing my screen so that you can see what it would actually look like to load the gel. Because like I said, it's just hard for me with my setup to show you the camera up close to my gel and have enough outlets and stuff. So while we're doing science from home, we have to do a little bit of pre-recording. So this is an overhead view of that same little gel box, and you can maybe see that there's some liquid over top of the gel. And that is this running buffer that I just mentioned. So that will help the electric current flow into the gel. And what you're going to see as I play this video is me loading some of this purple dye just into the gel. So I just put the pipette tip right into the pocket, and press down and then take it out. And then I get a new pipette tip and I put a new sample into the next well. Now on this one, you're going to see that there's a bit of dye stuck to the outside of the pipette tip. So it kind of floats away on the liquid. And that's just to sort of demonstrate, it's okay if a little bit of that happens when you're doing your practices. But if you're loading your DNA into the well, you want to make sure that all the DNA actually goes into the well. So it's actually inside your pipette tip and it goes into the well. But you also can't stab all the way down through the bottom of the gel. It's pretty soft like Jell-O, so it's pretty easy to accidentally stab all the way through the bottom. because you don't want the DNA to go underneath the gel. All right, so let me explain how the gel works and why we want that DNA in this little pocket. All right, so why does the DNA move through the gel? That's what's going to happen. I'm going to put this top on, and I would plug this in and turn it on, and an electric current is going to move through the gel. So why does an electric current affect DNA? Well, this has to do with the charge of DNA. So think back to that picture of DNA I showed you, that illustration of DNA I showed you at the beginning. Do you remember any parts of the DNA that might have had a charge, either positive or negative, labeled on it? Or maybe you've learned about this in one of our other labs. DNA has a negative charge, and it's from the phosphate groups on the backbone. So this is a slightly different illustration. You can see there on the outside, we have some negative charges. So when we apply our electric current to our gel, DNA is going to move away from the negative charge and towards the positive charge, so towards the positive electrode. I'm going to show you like a little cartoon illustration of this on the next couple of slides. I hope that makes sense. All right, so imagine this is our gel, and in this picture we just have one well. In our real gel we have a bunch of wells, so we can load multiple samples at once, but I'm just showing you one right here. And those little circles represent the molecules of agarose, so the sugar molecules. And they make up a matrix that fills up the space and makes it difficult for DNA to move through. All right, so here we have a blue pipette tip illustration and some orange DNA in my picture just for fun. And we get the DNA all into the well. Once we have all the DNA in the well and we put the buffer on top, we start the electricity. And so we have our positive electrode there on the bottom of my screen, far away from the wells. So no matter how you see the gel oriented, you want to make sure that the positive electrode is far away from the wells. That way the DNA moves away from the wells. If you think about in this picture, the wells are close to the top. If we ran the current the opposite way, the DNA would actually fall off the top of the gel, and that would be no good. So we want the DNA to move down towards the positive current. All right, so once that electric field is applied, the DNA is going to start to find its way through the matrix of the gel, through those little sugar molecules that are all lined up. Which pieces of DNA do you think are going to move faster? Do you think the big pieces are going to move faster or the small pieces? Well, think about that for a few seconds. And I'm going to advance it once. And now what you can see is that the small pieces have actually moved to the furthest away from the well. And there's big pieces that are kind of stuck up by the top of the well, by the top of the gel near the well. So why do you think that is? Well, you can think about it like a traffic jam. If there's lots of cars all lined up on the streets, something very small like a bicycle or a motorcycle could find spaces in between the cars and actually move to the front of the line, right? It might not be legal, but it could physically happen. A bicycle could move in between the cars and find a way to get to the end faster. Whereas a big vehicle, like a big truck or a big bus, is going to be stuck. They're not going to be able to find spaces in between cars. Likewise, these big pieces of DNA, they can't find spaces between the agarose molecules, so they stay closer to the well. All right, so the small pieces move faster, the big pieces move slower. So now we have separated our DNA by size. And now you'll be able to tell, first of all, how many different size fragments were in your tube if you had cut your DNA up with a restriction enzyme, and also how big those fragments are relative to each other. All right, so at the end, once we've stopped running our gel, we end up with a little accumulation of each size of DNA. And that's called a band. So it wouldn't be just one small piece of DNA like I have in this picture here. It would be lots of small pieces of DNA like in the previous picture. And they all accumulate where I have this little orange rectangle in something called a band. And we'll be able to see those bands when we expose the gel to a UV light or a blue LED light, depending on your stain, and they'll be kind of glowing. I'm actually going to show you a time lapse of a gel running on one of these little blue gel boxes. So these boxes that I showed you a minute ago have a blue LED light actually attached inside of them. And so you're going to be able to see on the next slide the DNA actually actively moving through the gel. If you use one of the gel boxes we usually use for our field trips, you won't actually see the DNA. You might see the dye. We mix some loading dye in with the DNA to help it sink to the bottom of the well. So you might see that blue or purple color moving through the gel. But in this particular video that I'm about to play, you'll actually be able to see the DNA, which is the glowing green stuff, moving down through the gel. Okay, so right now you can see that the DNA is all sort of up in the wells at the top of the gel. And as I play this video, you can see that dark colored dye is starting to move. So the dye also has a negative charge and moves in the same direction as the DNA. And now you can start to see small fragments of DNA on the right-hand side moving down. And over time, you'll start to see smaller fragments separating out from the other wells as well. Okay, so this isn't the most clear, but I think it's really cool that you can actually see how the DNA moves through the gel. And in the end, you would have different banding patterns in each of these samples. All right, so this is just an example gel for you. I'm going to show you our crime scene, and I'm going to show you what the gels look like from a class who did this lab. But that hopefully helps you see how the DNA moves through the gel. Okay, so remember, we're looking to match the pattern of those bands between our evidence and either our victim, suspect 1, or suspect 2. So here's our made-up crime scene. All right, so in this made-up crime scene, the police received a call that there was a gunshot in the middle of the night. And when they came to the house, they found that our victim, Jane Doe, here she's a little emoji, she was unfortunately killed. Okay, so they look around her house and they interview some different people and they come up with 2 suspects. Suspect 1 is John Doe. He's the husband of Jane, our victim. And suspect 2 is James Smith. He's a co-worker of Jane who had been at her house. All right, so I also want you just to take a look at this crime scene and see where I've put a number is a piece of evidence that we have DNA from. And again, remember, just because we find somebody's DNA in one of these spots doesn't mean that they were the murderer. It just means they left their DNA in that spot. Okay, and we also had some statements from each of the suspects that the police would use to sort of figure out who's telling the truth. They would think about who has a motive in this murder. But their short story is each of them blames each other, of course. Okay, so let's look at our first gel. And this picture got a little bit overexposed, but you can see I have from evidence 1, evidence 2, and evidence 9. And then our victim, suspect 1, and suspect 2 are run on this gel. So because this one's a little bit overexposed, it's a little bit hard to see that evidence 1 and evidence 2 look the same, but I'll tell you that they are the same. Now, if we compare those to the victim, and we compare those to suspect 1, and we compare those to suspect 2, which of those patterns matches evidence 1 and evidence 2? Well, I can see that it looks kind of like victim. We have one bright band and then a little gap, and then some more bright bands. It doesn't really look like suspect 1. Suspect 1 doesn't really have that gap near the top. And there are some bands that are smaller DNA that are further away from the top of the gel, the well, in suspect 1. And it definitely doesn't look like suspect 2 that has a lot more bands going down. So I think evidence 1 and evidence 2 looks the most like the victim's DNA in this case. Okay, now how about evidence 9? Evidence 9 has lots of bands. They go down a little bit further. They might be a little bit hard to see, but there are some bands. I'm just going to use my cursor. There are some bands down here that match suspect 2. Okay, so on the next slide, I'm going to have labeled evidence 1 and evidence 2 came from the victim, and evidence 9 came from suspect 2. All right, so evidence 1 and evidence 2, it turns out, are some blood that was found on the 2nd floor of Jane's home. So it makes sense that the victim has left her blood in many of these spots because she's the one who was murdered. So it makes sense that she was bleeding, right? So that's where that blood came from. It came from her. Doesn't tell us anything about the suspects, but it might tell us where she was at various times throughout the evening while she was bleeding. Okay, and then evidence 9 is from a knife that was found in the kitchen with some blood on it. And that has suspect 2's DNA on it. And the police had noticed that suspect 2 had a bandage on his hand. Mr. Smith had a bandage on his hand. So they asked him about it, and he said, well, Jane Doe and John Doe were fighting, and I was making us a snack in the kitchen. I got startled by their fight, and I cut myself. So that matches up to his story that his blood is on a knife in the kitchen. All right, let's move on and look at evidence 3 and evidence 4. Evidence 3 is some more blood that was found on the first floor of the home. And evidence 4 is fingernail scrapings. So if you don't know what fingernail scrapings are, this is like if someone had scratched another person or themselves and got some skin cells underneath their fingernails. Investigators could scrape those skin cells out and extract DNA from them. All right, so let's take a look at evidence 3 and evidence 4. So evidence 3 also has this little bit of a vertical line. And that's probably because the students who loaded this gel, it was their first time doing gel electrophoresis, and they stabbed just a tiny bit down into the gel instead of staying up in the space of the pocket. So some of the DNA kind of got stuck there. But that's okay, we can still see the banding pattern. And we can see that it doesn't really look like suspect 2. It doesn't really look like suspect 1. We don't see these bands down here. And we see a little bit of a gap here. So I think it looks the most like the victim for evidence 3. And how about evidence 4? Well, if you look at that pattern, I think evidence 4 looks the most like suspect 1. We have the same bands at the top, that little gap, and then 2 more bands down below. So let's label those. This blood on the living room floor came from the victim. Again, we're not really surprised that we find her blood in various parts of the home. And evidence 4, this fingernail scrapings from the victim, came from suspect 1, her husband. So that's consistent with the story also that suspect 2 said, which is that there was a fight between the couple, and she may have scratched him at some point. So far we don't really have any evidence that tells us who was the murderer, but we can start to piece together with their stories what may have happened. All right, let's look at 5 and 6. 5 and 6 are both pieces of evidence that came from our gun. 5 is some blood that was found on the outside of the gun. So perhaps someone who was bleeding touched the gun. And evidence 6 are some skin cells that came from the trigger of the gun. Okay, so that might be very informative. So here we can see evidence 5 and evidence 6. Now in this case, the students who ran this gel accidentally flipped suspect 1 and suspect 2 on the right-hand side. They flipped those 2 samples around when they were loading the gel. But that's okay. So we know from our other gels what suspect 1 and suspect 2 look like. So it was very easy to quickly identify that they had just made a mistake. And when I asked the students, they said, oh yeah, I think we loaded those in the wrong order. Okay, so no problem. But we can match evidence 5 now to suspect 2, right? We see those 2 bright bands at the top spaced out and more bands going down below. And then how about evidence 6? Does it look like victim that it's right next to? No. Does it look like suspect 2? Also no. That one looks like suspect 1, which is in this case on the far right-hand side. So we'll add those labels here. We have suspect 2, James Smith. His blood was on the outside of the gun. Well, we already know he was bleeding because he was cut. And then suspect 1, the husband. He's the one whose DNA was found on the trigger of the gun. All right, for the sake of completion, let's look at evidence 7 and evidence 8. So evidence 7, it's a little bit faint in this gel because maybe the students didn't get all of the DNA all the way into the well. Maybe a little bit of it floated out like you saw in my video of the loading dye. But if you look very carefully, there are some bands here, and we don't have this gap that we see in the victim's DNA right here. We see these spaced out bands just like suspect 1. So evidence 7 came from suspect 1, and evidence 8 pretty clearly came from suspect 2. So I'm going to add those 2 pieces of evidence to our crime scene. Now, I want to remind you that we don't necessarily know just from DNA what happened. Investigators would use the the suspect's statements. If the victim was still alive, they would use their statements and other pieces of evidence about their motives and their alibis if they weren't both in the home or they both didn't say they were in the home in order to actually solve the crime. But our DNA evidence gives some corroboration to parts of the story and may give more information like who was actually touching the gun, at least at one point in the night, okay? So maybe you can think about what you think happened in this story. Again, this one is just totally made up for fun for our classroom use, but that is how we would use DNA. We would match up whose DNA was in each piece of evidence to try and provide some more information to investigators. All right, so I hope you have enjoyed this day of DNALC Live so far. I want to encourage you to check out our other DNALC Live videos. They'll all be streamed here on YouTube. You can also go to our website, dnalc.cshl.edu, and see links to all of our DNALC Live content, where we're sharing lots more information about things like restriction enzymes or other enzymes, about gel electrophoresis, and all sorts of other great molecular genetics topics. So I hope you'll check those out. If you are a teacher or you know a teacher who might want to book a private field trip with one of us to also have a chat with your classroom, you can also look in our online programs. We have virtual lab field trips available. And once again, at the bottom, I have all of our social media channels. I encourage you to follow us, subscribe to our YouTube channel, turn on alerts so you don't miss any of the videos that you might be most interested in seeing. And if you have any particular topics you want us to cover, especially if they fall in the area of genetics or biology in general, let us know. Send us a message on social media, leave us a comment here on our YouTube video. If you have any questions about this video after the live stream ends, you can also leave those in the comments and we will do our best to come back and answer them. So that's it for me today. I hope you all have a great day and come back and see us again on DNALC Live.